This algorithm that we've just put together is sometimes referred to with the very long acronym UPGMA, which basically means that we're taking the average of the pairwise distances. The point being that once we've joined a set of sequences together over here, and we've joined a set of sequences together over here, now we want to compute the distance, if you will, between this cluster and this cluster. It's just going to be the average of the pairwise distances between them. So, sort of long-winded way of saying it's just the average. Okay. This algorithm, I think you can guess that it gets used not just in phylogenetic tree construction. It's a general hierarchical clustering algorithm that you can apply on any distance function. So you've got some set of things, you have a distance function that can compute pairwise distances between them, and you can apply hierarchical clustering. People do this on expression data, for example, and they do it outside of bioinformatics as well. But it's the same extremely simple algorithm. This UPGMA, I think you can see that it perfectly fits the ultrametric distance assumption. Basically, to get the distance from these sequences over to those sequences, the best estimator is actually the average of those, since there's random noise for any individual pair. And let's see. We can use this also to compute the distance for the MRCA based upon basically splitting the distance between this group and this group to get the actual distance going back here. We can then use that to compute the distance for internal edges. And the whole algorithm can be written in this sort of trivial pseudocode. You have an initial list where it's just every single cluster is one single sequence. And then you simply have an iterative cluster joining algorithm where you have distances for all possible pairs of clusters, and choose the closest pair. You join that pair, and you replace those two clusters by a new cluster, basically using the averaging to compute its distance versus all other possible clusters. Repeat, 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 until all clusters are joined. Question?
Is that distance that you compute useful for anything, or is it just to recover the
Well, you could think of it as becoming a better and better estimator of the time, going back to the MRCA of the set of sequences that you've now clustered.
Okay.
One way of thinking about it. This is sort of superfluous, but you know, if you have some example data here, it gives you a table of pairwise distances. Here, the distances are computed just trivially as just counts of mutations, but for a real phylogenetic construction, the distances would actually be computed in a much more sophisticated way based upon estimating delta AB under a specific transition matrix, instantaneous rate matrix. But just to follow the obvious algorithm, we look at that table, we see a minimum distance. Mac versus orang, or chimp versus human, both have distance of 2, so we end up joining. That's the whole algorithm. Okay, so